Hi again, and thus far we've talked about the structure of cells when it comes down to cells, the function of the organelles, but we really have three more important topics and things we need to talk about in regards to cells, okay? One of those is cell division, which we'll be getting to very shortly. Another one, which is going to be really important as well, is DNA and RNA. But today, what we want to do is we want to talk about one of the more important things in regards to cells that I think we all need to have a good handle on, because it's going to be coming up time and time and time and time and time again down the course of the rest of the season or semester. And basically this is the function of the cell membrane and transport of things across that cell membrane. Um, it's sort of a deep subject, so be ready. Hope you have a little time to sit there and think, because we're ready to get going here. So let's talk about cell membrane and how it works and how we get things across the cell membrane, because this is, like I say, a very, 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 very important subject we're going to be talking about many times during the rest of the semester. Well, first of all, we talked about this before when we briefly discussed the cell membrane. Is basically it has a selective permeability. Okay, it only likes certain things to get in. We can't let everything in. This selective permeability is really important, because there are things on the outside that we just don't want in, but there are things that we want to get in, and we want to open the doors for the things that we want to be able, the cell to be able to use. Okay, so the cell has to have a way to be able to communicate between its internal environment, which we call intracellular. Okay, so intracellular, I-N-T-R-A, cellular, means inside the cell, and extracellular would be outside the cell. So it has to be some way to communicate what we need, okay? Also, we find that the cell membrane provides what's called an electrochemical gradient. What I mean by this is the outside of the cell might be charged one way, and it's usually charged positively, okay, where the inside of the cell membrane is charged a different way, which means it's charged negatively, okay? And we'll talk a little about that and the importance of that. That will become exceptionally important when we talk about a muscle. We'll talk when we talk about a nerve conduction, and those things, this membrane polarity, or this electrochemical gradient between the inside and outside of a cell, is exceptionally important in those areas. So this is what we're going to be talking about during the course of this next of this video. Okay, first of all, just as a review, and hopefully this is something that you know from when we talked about our cell structure in the cell membrane. We know that it's a lipid bilayer, and it has a couple different lipids. This is actually the previous slide that we had from the first lecture. We know that it has three layers, but the most important one is supposed to be this phospholipid. Okay, most of the proteins that we have are called glycoproteins. Now, there are a number of proteins that we have, and we showed these before, and today we're going to be talking a lot about what those proteins do, because not all proteins that are in the cell membrane are the same. Some are going to do different things, and other ones will do totally other things. Okay, and we talked about these proteins basically are like proteins that float like icebergs in a sea of lipid. Okay, now these proteins can be what's called transmembrane. Trans means across. If you look at this thing right here, it's this one, right? It's going across the membrane, which means it's probably going to be carrying something or allowing something to pass through a pore, whatever the case may be. We also have what are called peripheral. Okay, and the peripheral ones, like out in here, and some of these are receptors that will identify a certain material that's in the extracellular environment that may allow something to happen within the cell membrane, particularly when we talk about things like insulin. Okay, and also we have some that are integral, which are actually proteins that are within the membrane itself. Okay, so the proteins can actually act in a variety of different ways. Okay, which is what the hardest part of understanding what this lecture is all about is going to be entailing. So proteins actually have a purpose, and these proteins have multiple different purposes. Okay, well, first of all, let's talk about some of these plasma membrane proteins and what they do. First one we have to talk about are what are called ion channels. Okay, and let me get rid of my picture down here so I can see all this stuff here. Ion channels, and ion channels are basically the channels, or they're pores, that allows a specific ion to move through the membrane. Okay, and if you look up here at the top, it looks like it's like a boat going down a canal from one body of water to another body of water, goes to a canal to get from one area to the other. And what happens is these ion channels allow a specific ion, okay, to move through the pore. We'll talk about this over and over and over again during the course of the semester about these ion channels. There are many different things that will move across through these ion channels. But basically, it's a pore. It's a pore and allows things to move through that pore at the right time in the right place. So that's what we call an ion channel. We also have what are called transporters. And transporters are basically proteins that will pick something up on one side of the cell membrane and transport it to the other side, such as amino acids. You know, we know that when proteins are made of amino acids, so I need these amino acids to make more proteins, and some of these amino acids might be in the extracellular fluid. So I have to get them inside the cell so the cell could use these amino acids to make a protein. How do I get them in there? Frequently by transporters. These proteins will pick up this amino acid or another substance on one side of the membrane and actually transport it across the other side of the membrane, or body surf it across, more like, okay? We also have what are called receptors, okay? And like I mentioned, these receptors are on the surface. They're usually a peripheral type of proteins, and what they do is they recognize and bind specific substances that will alter cell function. Typical example, glucose. Glucose is an exceptionally simple molecule. It's only C6H12O6. It's not a huge molecule or anything like that. But on the other hand, it doesn't get in the cell easily. It doesn't pass through this cell membrane easily at all. In fact, it can't. It has to go through some other way. And so what happens is when we want to get glucose into the cell, and using the glucose for an energy source, what happens is the pancreas releases a hormone called insulin, okay? And that insulin gets transported throughout the body, and what happens is it's in the bloodstream, it gets transported around, it gets into the area around, gets out of the blood vessels and gets into the extracellular fluid and looks for cells. On the surface of cells, there will be a receptor for insulin. Once that insulin binds to an insulin receptor, which is a protein on the surface of the cell, then what happens, it allows a process by which glucose can now go from the outside, the extracellular environment, inside the cell, or the intracellular environment. So that's what we call a receptor. Another example of that would be something called ADH. ADH is a hormone that's produced by a small little portion of the brain called the pituitary gland, sort of about the size of a pea or the tip of your little finger. It's in the base of the brain, and what happens is when I want to conserve water, what will happen is the pituitary releases this hormone called ADH, antidiuretic hormone. And in certain area in the renal tubules, in the distal convoluted tubule and the collecting duct, which I don't know if you remember right now, we'll talk about at the end of the semester. What will happen? There'll be receptors that will bind for this ADH, and once the ADH binds to the receptors, it allows water to be sucked out of these tubules and back into the circulation to conserve water. So these are called receptors, okay? And that's a third type of a protein. A fourth type of protein we get are called enzymes, and frequently these are either on the inside portion of the cell membrane or on the outside, okay? And what we talked about, these enzymes actually catalyze reactions, you know, either inside or outside the cell. So in other words, they'll actually make a chemical reaction to occur, just like yeast will cause bread to rise and stuff like that. These will actually catalyze a particular reaction. Again, if you see a word that ends in ase, chances are that word that ends in ase has something to do with an enzyme. Okay, so that's called, and these are enzymes, and enzymes are proteins. That's my fourth type of protein. My fifth type of protein are cell identity markers. Cell identity markers. Now these are really critical. Okay, even though, like I mentioned in the very first lecture, that we're all exceptionally similar to everybody else in the class, we're all exceptionally different. Okay, and the way we're exceptionally different is each of our DNA is ours and ours alone. It's special. The only time you might share some DNA with somebody is if they're an identical twin. Okay, so what happens is there are certain protein markers on the outside of our cells that are based upon who I am, and they have certain code as this is me and nobody else. Okay, and this is called the major histocompatibility complex 1, or MHC1 a marker. And this MHC1 marker is basically has the code for who I am, which means that if I was to receive tissue from somebody else who has a different major histocompatibility complex one marker on the outside of their cell, my body would reject it, okay, because it's not me. So this is sort of like these are cell identity markers on the surface of the cells that say that this cell is mine and mine only. There's nobody else that has a cell unless it's an identical twin again, okay? And these are called cell identity markers. It's major histocompatibility complex 1 or MHC1 markers that are on the surface of the cell, which are proteins, okay? The sixth type of a protein we have are cytoskeleton anchors, okay? And this actually allows the filaments, like we talked about before in our previous discussion when we talked about organelles, to be able to anchor it to the cell membrane. So the cell membrane retains some shape and structure, okay? So it provides stability to the cell itself. So these are some of the plasma membrane proteins that we have to know about. Ion channels allow an ion to pass through the membrane. Transporters, or proteins that will carry things from one side of the membrane to the other side of the membrane. Receptors that are sensitive, like a lock and a key, that will be receptive to a particular substance that allows another action to allow something to be taken inside the cell. Enzymes, which catalyze or produce some type of a chemical reaction on either the inside of the cell or the outside of the cell. Cell identity markers, which actually distinguish who I am, okay, as well as cytoskeleton anchors or linkers, basically, which provides the cement that holds these microtubules and stuff like that that'll give the cell its shape and structure and stability. Well, what happens is about the membrane, like I mentioned before, is one of the important factors is something called selective permeability. Selective permeability. Things are able to move through the membrane if and only if they meet the right characteristics, if they're the right things, if they're the right materials, you know. If you look at the people there trying to get past the doorman, maybe they look for the pretty people, you know, the people that are good. I never get in anything like that. But anyway, so there are a couple factors on a chemical basis that may influence what gets in and what gets out. Is the lipid solubility of a substance. We talked about this before, is because we have that lipid bilayer. That lipid is not agreeable with everything. It doesn't like the water. That's why we have something that's hydrophilic on both sides of that lipid layer to make it compatible with the intracellular environment, which is fluid, and the extracellular environment, which is fluid. But that lipid bilayer is sort of like a barrier, okay? So it depends upon if something's really lipid soluble. If something is able to dissolve in lipid, it's probably going to have a better chance of getting in. It's more compatible with it, okay? Also the size of a substance. Think about this. The larger the substance, the more difficult it is going to be get through some type of a barrier. Just logical, okay? So the size of a substance may make a difference. The charge of a substance. Again, the charge, you know, if you remember when you're a kid, you had a magnet, you know, and you always like to take the magnet and you put a positive and a negative pole, and basically the thing stuck together on the magnet. But then if you took those two positives and you put the two positives together, they'd repel each other. So as a result, the charge of that substance might make it a lot more difficult to be able to get through that membrane, depending upon what that charge is. Also, the presence of specific carriers or pores. Okay, like we talked about, these protein things that will either, as a pore, allow things as a channel to come through, or a carrier that's going to be able to pick something up and carry it across the membrane. So and again, there might be some cells that have a lot of protein carriers or pores, and there's some cells that might have very few protein carriers or pores. So all those things would make a difference. Okay, so this is something that will help to influence selective permeability, getting in there. Again, stop. Okay, you can't get in. You are but you can get in. Okay, I remember years ago going to when we were in California with my wife, going to a TV screening for a comedy show, and basically what they did was they want to have, you know, you'd be sitting in the audience and stuff like that for that, and as you walked in, they had a little guy that would stand behind after you walk past the door, and he'd hold up 1 finger, 2 fingers, 3 fingers, or 4 fingers. 1 finger meant that's a number 1, that's a good one, that's one of the beautiful people. Number 2, well, they're okay, but they're not quite so good. Number 3, that it's there, maybe let's sort of put them somewhere where we don't want them to see. And number 4 is the face that only mom could love. Okay, so basically what they did is, as they went through, this guy would hold up his finger 1, 2, 3, or 4. If you're a 1, they put you in the front row, which meant that you got in the front of the theater. If you were a 2, they put you in the second row, which means that you were still there, but you might be seen. If you were a 3, you weren't going to be seen. They're going to put you in the shadows in the back of the. And 4, man, they kept you in the bathroom the whole time for the whole string and said, "Oh, you missed it? Where were you? You were in the bathroom." They didn't want you to be seen at all. So anyway, and so this is what the membrane does. It is sort of selective as to what it wants to get in. Okay, that's the so there's probably a little guy. There's a little. I'm gonna propose another type of a protein carrier that will actually stand there when the material comes to the surface of the cell. It's either gonna go to 1, 2, or 3, or 4. Okay, and I think that would probably work as well. So anyway, that's selective permeability. Things get in, other things don't get in very well. Okay, more difficult. The cell likes to admit only the right stuff. If it's the right stuff, fine, you're in. If you're not the right stuff, man, it's going to be a pain to get inside. That's probably a good thing in some ways. So, what is the membrane permeable to? Okay, first of all, because it's lipids, the lipid bilayer is permeable to things like nonpolar uncharged molecules. You know why? Because they don't irritate anybody. They're sort of like just, they get along with everybody. Nonpolar, they don't really have a charge, stuff like that. And they're also, small things like oxygen, carbon dioxide are small molecules. Steroids are larger molecules, but steroids, guess what they're mostly made of? Fat. Okay, and so as a result, because they are more lipid with the steroids, they're more compatible with that lipid bilayer. Okay, fat mixes with fat. Fat likes fat. So anyway, that's what's happening with that. So that's what's more permeable to. Lipid bilayer is impermeable to things like ions or charged or polar molecules. Okay, why? Because again, that charge is sort of irritating. It's not what they want. It's sort of like, I get that guy. He keeps on. Every time I go by him, I get a shock. And things like glucose. Glucose is a relatively small molecule, like I mentioned, but it is polar, and as a result, it's very difficult to get. You can't get through. It has to be. Remember we talked about the insulin, how the insulin has to actually say, I vouch for the glucose to get the glucose inside the cell, because otherwise the cell doesn't want the glucose in. And glucose is a great energy source because it's a clean burning fuel. When we burn glucose for energy, basically the byproduct is carbon dioxide and water. Easy to blow off or exhale the carbon dioxide, easy to urinate out the water. Okay, the lipid bilayer is slightly permeable to things like water, urea. Urea is a nitrogenous compound, mostly a waste compound from the breakdown of proteins. And what happens? These are relatively small molecules, and they usually are able to fit and work through these small little gaps. Okay, so basically, this membrane is sort of like customs agent. I don't know about you, but every time I've gone through customs agent, you sit there and you sort of shake. And I could tell you one story a long time ago. We went to. I was doing some lectures and helping to set up a lab in Bogota, Colombia, of all places. Okay, and I took some supplies down with me. Okay, and one of the supplies I took was actually a methyl methacrylate, which is it's an acrylic material you mix with a solvent, which now you couldn't do because the solvent would be flammable. But at that time, they didn't care what you put in your luggage. You could have a dead body in your luggage. And so anyway, I get down to Colombia and I realize, here I am. I'm in Colombia. I'm going through customs, and they have this little traffic light whether red or green, and you pressed a button. If it came up green, they passed you on through. They didn't even look at your bags. If it came up red, man, you were almost like strip searched, and taken in another room and put in jail for 3 days until they could verify that you were in. So all these people are in line. I realized that the stuff I took with this methyl methacrylate was in a white powder. Duh, I was really stupid. Going to Colombia with a white powder in a bottle? Oh God, give me a break. And so I'm getting closer, and everybody's pressing the button, and all these green lights coming up. I said, oh my gosh, I'm gonna be a red light. And I'm sitting there, I'm sweating. I probably look like the biggest criminal in the world. I'm sweating. I'm like jittery and nervous. I pressed the button. I kept my eyes closed. So the guy says, hey, go through, whatever he said, vamoose or whatever. I don't know. Anyway, so he got me through, and I pressed a green. But I sort of like made it. Luckily that time. But it's like customs, okay? Or the three little pigs, no, you can't come in, not by the hair on our chin, you can't come in. So that's sort of like some things are not going to be able to get in the cell. Okay. So then, how can we get stuff in? Okay, now these are a number of factors that will allow whether things get in or whether they don't get in. We talked about a few of those things and presence of proteins and all kinds of other stuff. So let's talk a little bit more specifically and get a little bit more in detail about how things do get in, how things don't get in, and what we prevent from getting in. Okay, first of all, transmembrane proteins. Okay, we talked about these transmembrane proteins that have a pore. Okay, and what happens? They act as channels to allow a variety of small and medium-sized, sometimes polar, which might be charged, charged substances that don't normally pass through the membrane itself. Again, polar molecules and charged particles, or ions, they don't get through the membrane, okay, because the lipid bilayer is impermeable to them. However, if there's a special door that they could get in, a special pore, those things could get in. And we're going to see this time and time again in regards to sodium and potassium with muscle contraction and with nerve conduction, stuff like that, as we talk about those later on down the road. Well, what these pores are, they're like a door, like the cat door up at the top. Cats, yeah. Anyway, they're a cat door. And they allow things to get in, okay? So they're selective as to what they transport. In other words, certain pores will be selective to allow sodium. There are sodium channels that allow things to get in. There are calcium channels that allow calcium to move and stuff like that. So these are selective as what they get. Macromolecules do not pass through. So macromolecules, so this cat could get through, but in these macromolecules, you know what? They can't get through. So luckily that bear couldn't get through that small little door in the in the small little cat door in the door. Okay, so macromolecules have to get either in or out when they're very, very large by something else, which is called endocytosis, which means taking something from the outside, wrapping it up in a package and pulling it, and that package actually fuses or merges with the cell membrane, and then what happens is it goes through as a large package and it opens up on the inside and dumps it. Or exocytosis was just as the opposite. When they get rid of waste, they put them in a big bubble, and that bubble goes to the cell membrane surface on the inside and merges or fuses with it, and then all of a sudden it allows it to spit it out to the outside. That's called endocytosis. Endo means within, getting things in, and exocytosis, exo means without, putting things outside the cell. So that has something to do with these transmembrane proteins that will allow that, or the transmembrane proteins. If the material can't get in through a transmembrane protein, then it's going to go exocytosis or endocytosis. Okay. Now, membranes, I mentioned, also have what are called membrane gradients, which means that it keeps various substances on different sides of the membrane, again, because of what it's permeable to and what it's not permeable to. Okay, and there are two ways that we look at that. One way is called a concentration gradient. And a concentration gradient is that what happens if we look at the concentration of certain ions, they're in a higher concentration on one side of the cell membrane as compared to the other side. Now, if we look at sodium and potassium, so sodium is a very high concentration in the extracellular fluid or outside the cell membrane. Potassium is a higher concentration on the inside the cell, okay? Well, you're going to say to me, but Dr. Litchik, aren't those both cations, or cations we know are positively charged ions? Yes, they are. Well, if I look at the cell membrane, the outside of the cell membrane, if I look at the outside of the cell membrane, the outside of the cell membrane is more of a positively charged. The inside of the cell membrane is more of a negative charge. Okay, and the reason why it is, and that gets to me, my second thing is my electrochemical gradient. And what happens is, I keep a lot of sodium on the outside, so that's a concentration gradient. I have less sodium on the inside. If you take blood out of somebody and you look at their sodium levels in the blood are very high. If I look at potassium, the potassium levels in the blood are very low. And the reason why is most of that potassium is sequestered inside the cell, which is not amenable to be able to get in the chemical or in the liquid portion of blood, because it's inside the cell, intracellular, where outside the cell in the extracellular fluid of the plasma, sodium is abundant. Okay, so you actually read that. So, but sodium and both potassium are both cations, they're both positive. Okay, but so I have sodium on the outside, which is good, and a small amount of sodium on the inside, large amount of potassium on the inside, and a small amount of potassium on the outside. But we also have what's called this electrochemical gradient, and the electrochemical gradient says I have a lot of sodium on the outside and potassium is cation on the inside, but on the inside of the cell membrane, I have also a lot of things which are basically provide this electrochemical gradient because what I have is a large number of proteins on the inside. So therefore, the inside of the cell membrane would be more negatively charged, and the outside of the cell membrane would be more positively charged. Positive on the outside because the sodium can't get in, okay, very well. Only could go through pores at the right time. So I have a lot of sodium, which is a positively charged ion on the outside of the cell. And the inside of the cell, I've kept potassium in, but still positively charged, but I have large molecules like proteins, which have a negative charge. So therefore I have a large amount of proteins, a large amount of negative charge, which creates that the inside of that cell membrane is more negatively charged and the outside is more positively charged. So in both these situations, we've had both a concentration gradient, a lot of sodium on the outside and not much in the inside, a lot of potassium on the inside and not much outside. So it's a concentration gradient. And the beginning of the concentration gradient, because I have a lot of sodium on the outside, which is positive charged, I have a lot of negatively charged materials like large proteins on the inside, which creates this electronegativity on the inside and electropositivity on the outside of the cell membrane. Okay, hope that's explainable to you. And this electrical potential, as well as this concentration gradient, is going to come up time and time and time again. Okay, when we talk about, again, muscle contraction and nerve conduction, particularly, that's going to be the most important places where we're looking at both the concentration gradient and the electrochemical gradients. And also with the concentration gradient and electrochemical gradient in the cardiac muscle too. That's also very important there. So hopefully you understand a little bit about that positive and negative. Sort of like, like a castle. The castle wall keeps certain things, there's a gradient, on the inside as regards to concentration as well as electrical charge. Biotransport is another word that we should probably know, and biotransport is basically just another method of getting things through the membrane in one manner or another. Okay, and this again requires us to understand what's going on with cell membrane structure and function, like we talked about before. So that's a little bit. We'll talk a little bit more about biotransport. So in other words, methods of getting things in a little bit more, and we'll talk about some of the factors a little bit more specifically in regards to that, as well as methods of getting things out. Okay, so let's get on and talk a bit more about biotransport. Now, biotransport requires a number of different possible scenarios. Okay, and we talked about gradients and stuff like that, and pores and how things get in. However, there are ways that we could get cell in chemically. I think our materials in chemically: osmosis, diffusion, facilitate diffusion, active transport, and transport in vesicles. Okay, let's talk about each of those. Let's talk about each of those all together. Let's first talk, and we'll talk about all those. Okay, a couple things that we have to think about these things though in regards to all these processes is that some need energy and some don't. Osmosis, diffusion, and facilitated diffusion do not need energy. Active transport needs energy. Now, the biggest thing I want you to remember here, the word active transport. If you see active transport, it means for that to happen, it requires energy to be able to be produced and released to be able to move things across. These things up here, osmosis, diffusion, and facilitated diffusion, don't need energy. Active transport needs energy. These don't need energy. Active transport does need energy. Okay, and that's going to be a little bit more apparent as we go down the road here. Okay, let's talk about passive transport. Okay, passive transport basically doesn't need energy. Okay, because active does need energy, passive doesn't. There are four main methods that I could get things across the cell membrane by passive transport. Okay, one of those is diffusion. Okay, another one's called facilitated diffusion. Another one's called osmosis. And filtration we won't talk about now, but that's something we use that's more related to the kidney. We'll talk about that later on, but we will talk about diffusion, facilitated diffusion, and osmosis. Okay, and let's talk about those individually. Okay, and this is just sort of a diagram to show you, diffusion through a channel, facilitate diffusion, and then active transport. Let's talk about those individually. Okay, so passive transport requires no energy. Okay, so like that lazy river and just floating down a river, and you don't have to paddle, you don't have to swim, and just sit in these little tubes, get sunburn, and that's all you need to do. It's very passive. Okay, let's first talk about diffusion. What does diffusion mean? Diffusion is a random mixing of particles. Okay, now when something diffuses, it depends upon five significant different characteristics. Five characteristics. Number one, the concentration gradient. Okay, the greater the difference in the concentration from one side of the membrane to the other, the greater the degree of diffusion. Okay, the idea is we all want to get to a point of what's called equilibrium, where it's balanced. Okay, like we talked about in probably our first discussion so long ago. Okay, everything wants to be balanced, and that's basically, we talked about homeostasis as a balance. And this concentration, if I have a large concentration of something on one side of a wall and a lower concentration on the other side of the wall, and that stuff could get through the wall by one way or the other, by a pore, by a transporter protein, by whatever the case may be, it's going to go from it's always going to go from an area of higher concentration to an area of lower concentration. It makes sense. Okay, if I have more stuff on one side, I'm going to give more to make it equalize out. More of the stuff is going to go from the areas where there's a higher concentration to an area that's lower concentration. That makes a difference. So the greater the difference of concentration, the greater the degree of diffusion. So let's say if I have one particle on one side and I have 9 particles on the other side, and I want it to be balanced, okay, I'm going to want to get 5 and 5 on each side, because now 1 plus 9 is 10. Okay, so I have 10 particles total inside and outside. I want 5 on each side. If I'm able to get that material across that membrane, either by a transporter, by simply passing through without a pore, or through a pore. Okay, what's going to happen is it's going to go from the higher concentration where there are 9 particles to the part of 1, until we finally get to that equilibrium where there's 5 on each side. So the concentration gradient makes a difference. The second thing that makes a difference is temperature. Okay, and what temperature does, the higher the temperature, the faster things move, and the faster things move, the more motions there's going to be able to get from one side to the other. So temperature makes things move faster. If you heat something up, things happen faster. Okay, that's just what it happens. Okay, the mass of the diffusing substance. And we talked about this before. The larger the particle is, the longer it's going to take to diffuse. The smaller the particle is, the less resistance it is. Take about 5 people and stick them in a swimming pool, have a race from one side to the other side, and one guy is 120 pounds, lean, mean fighting machine. The other guy in lane 4 is 640 pounds and takes up a lane and a half. Okay, which is going to get to the other side of the pool faster? I'm betting on the guy that's skinnier, because there's going to be less water resistance as it goes through. Okay, so the mass of the diffusing substance, the larger the mass, the slower the diffusion rate, simply because there's going to be more resistance. We also talk about the surface area. And what I mean by the surface area is, if I'm able to diffuse over a larger area, there's going to be more diffusion. But if that area that it's only able to diffuse through is a very small area, it's going to be harder to get those things through a smaller area. It's sort of like if you have 50 people in a room and they all want to get out of the room at one time, but one small door, guess what's going to happen? There's going to be a bottleneck at the door. It's like when you're driving into school and the highway has a bunch of orange barrels on there. That should be normally moving, because I have 5, maybe 4 lanes across. But if that 4 lanes is down now to 1 lane, what happens to your speed? Your speed is slower. So as a result, what happens is the surface area, if I had 4 lanes open, boom, the traffic goes through. If I have 1 lane open out of 4, everything narrows down and things get slower. So the larger the surface area, the greater the diffusion. Okay, so the larger the diffusion surface, the greater the diffusion. Also the diffusion distance. Okay, the longer it has to diffuse, okay, the longer distance it has to go to diffuse, the longer it takes. Okay, this becomes clinically significant in people with like a chronic obstructive pulmonary disease. What happens is the areas of the lungs become scarred, so to get the oxygen from inside these little air sacs called alveoli into the blood has to go through a longer distance, it has to go a longer way to get from, because of all the scar between around these little air sacs, from the inside these little air sacs, alveoli, into the blood vessels. So therefore it takes longer. That's why their blood oxygen levels are lower. Makes sense. Okay, so concentration gradient, temperature, the size of the diffusing substance, mass, the surface area over which they can diffuse, and the diffusion distance all make a difference. Okay, in regards to how things diffuse. Here's a diffuser. Okay, obviously some tea. Okay, Heat up that tea, heat up hotter water, guess what? Boom, it's going to go faster. So let's talk a little bit about this diffusion again, okay? And diffusion just means a random mixing of these molecules or ions, okay? And again, it's due to their what's called their kinetic energy. You know that all molecules are moving to some degree. So they all have motion and things are in motion. So what's going to happen is, let's say I have, if you look at the and again, we go until equilibrium is reached. If I look at the model below, this is where we start out here. So we're starting out at this area. You see all these particles, okay? This isn't that extracellular fluid, okay? The extracellular fluid is loaded with a bunch of material, okay? Whatever it is, okay? But what happens is this extra, we're going to, and let's say that these materials are able to diffuse through the cell membrane wall. Here's the lipid bilayer in here, okay? And there's the lipid portion in here. So let's say these things go through. They're going to go through. Eventually what's going to happen, if I counted the number of particles on this side to the number of particles on this side, they'd be the same. I hadn't counted, so I'm assuming it should be the same. And that means that they've reached an equilibrium. And what's going to be those factors? Concentration gradient, the temperature of what's going on here, the size of the particle, the area across which it could diffuse, as well as the distance, okay? And basically that's what we see with diffusion, this random mixing, okay? So like a pool, boom, those balls are going to go until they stop in motion, and they just randomly mix throughout the table. Now, again, when we look at how things diffuse through a membrane, it's not always the same, okay? We talked about what happens is nonpolar molecules like oxygen, carbon dioxide, some nitrogen gases, fatty acids, again, fats because the lipid likes fats, okay? Steroids are mostly lipid, some fat soluble vitamins. Oh, all these things have, a lot of these things have something in common. They're basically fatty. And so as a result, because of the fat area here, the lipid in the middle, okay, those lipoproteins are, what happens is gonna these things get through, are able to diffuse, and that's just that general diffusion like we showed in our last image, okay? So that's just the general diffusion which we saw before, okay? Then we could have things that could actually go through these ion channels like we talked about. Some typical examples are sodium. We're going to be talking a lot about sodium channels, potassium, chloride, calcium. What happens is on muscle contraction, the first thing that happens is when the cell membrane becomes stimulated, it opens these sodium pores to let the sodium go from the outside to the inside. Therefore, the outside becomes a little bit more negative because I'm losing that positive charge like we talked about before. So the outside becomes a little bit positive, it becomes more negative from being positive in the beginning. So what happens is potassium's on the inside, and potassium says, hey, you know what? I know it's getting a little bit more negative out there. Let me go out there and take the place of the sodium, and the potassium rushes to the outside. And that happens time and time again. We'll be talking about that. These are called gated channels. These are called gated channels, which means that here's a gate. There's a gate right down in here, and that if that gate's open, what happens is things could go through. But what happens is basically now the gate's closed, so things can't come through. So we'll talk a little about gated channels in a second. So the channel can either open or close. Again, in muscle contraction, when that cell membrane, or when that sarcoplasm, which is, the sarcomere, which is the cell membrane of a muscle cell, becomes stimulated, it opens the gate to let the sodium rush in. Same thing happens in heart muscle, okay, with muscle contraction and heart muscle contraction. So that's a little bit that. Then we also have facilitated diffusion. And facilitated diffusion is, again, when we talk about have a carrier protein, and what it does is it picks up the material and carries it across the membrane. Again, that's with glucose because now insulin's opened a receptor or bound a receptor, open the door to let that carrier protein take the glucose from the outside inside. Also other sugars like fructose, galactose, also some vitamins and stuff like that, and gets across the cell membrane that way, okay? And basically, oh, do you remember that? Red Rover, let somebody, whoever your name is, come over, sort of like we're able to get through, but it's sort of a game at that point. This is a gate, just sort of like an idea we're talking about gated diffusion. This guy, he wants to let some of the sheep in, so he opens the gate, and once he closes the gate, guess what? The rest of the sheep can't get in. Okay, so that's called gated diffusion. We see that with like sodium, we see that potassium, calcium, chloride, and things like that. Okay, that's called gated diffusion, and we're going to be talking about that multiple times during the course of the semester. Facilitate diffusion, again, is the passive movement via transmembrane transporters, okay? And these transporters basically are going to carry something across like we talked about. So passive movement, it does not require energy. via the transmembrane proteins, and they act as transporters. Okay. Again, some of the maximum diffusion rate also depends. Another factor that we have to think about is the number of available transporters. In other words, if the cell membrane only has a certain limited number of transporters, I'm only going to get a certain limited amount of stuff across that membrane. When do we use facilitate diffusion? With polar or charged particles. Because why? Because that lipid biolayer does not like them. It doesn't like to get through that. Again, glucose, fructose, galactose, and some vitamins can go in through facilitate diffusion. Okay? That's it. Now, this is a little bit more difficult to understand for the time being. Okay? And this is called osmosis. Okay? Now, osmosis is, before we even talk about osmosis, there are two words I need to know, okay? One word is solvent, and the other one is solute. Solvent is basically the fluid. If I take two things, if I take a glass of water, put a glass of water out, and I'm putting a package of lemonade in that, I put the powder inside. The water would be the solvent. The lemonade powder would be the solute, or the particles that are put in. So remember, solvent is the fluid, solute are the particles, okay? Solvent, the fluid, solute, the particles, okay? Remember that. That's going to be really important to understand. So basically what osmosis is, well, everything we've talked about thus far has been the movement of some type of a particle, such as, sodium, potassium, glucose, stuff like that through a membrane. What we're talking about here is the net movement of a solvent through a selectively permeable membrane, okay? So what happens is that solvent in the human body is none other than water, okay? In living systems, the solvent is water. And what I mean by this selectively permeable membrane is a membrane that only lets water through. It keeps everything else divided, okay? So what happens is, if I have a selectively permeable membrane that's permeable only to water, and water is a solvent that can move through the membrane, what's going to happen is water moves by osmosis from an area of higher water concentration or a more dilute state to an area of lower water concentration or to a more concentrated state. Okay? Until when? Until an equilibrium is reached. Okay, we'd also say it goes from an area of lower solute or particle concentration, which also means more water, more dilute, to an area of higher solute concentration, which means there's less water and more concentrated. Now what we see up in this area right here is, let's say we have two vats, and let's say these two vats started out the same level. Okay, so let's say that this vat over on this side, on the right side, was up to this level, and the vat on the left side was up to this level. But this is the semi-permeable membrane right here in the middle, and it's only permeable to water. The idea is to try to get the concentration on both sides of that membrane equal. But the only thing that's able to pass through that membrane is water. How do I get that concentration equal? The water now is going to run from this side over here to this side, which means that that water level is going to lower because more of it's going to go over to here to raise this up to make the concentration of this fluid on this side equal to the concentration on this side. And that's only because water is able to go from an area of lower concentration or more dilute area to an area of higher concentration or more concentrated area. And it finally is going to continue, and this level will rise and will rise and will rise on this side until now the concentration on this side will be the equal to the concentration on that side. Okay, and that's called osmosis. Okay, and this happens it's a way to move water from one compartment to another compartment, and it's by osmosis. So hopefully you understand that. Again, this compartment on this side was higher to begin with because there was more water. It was more dilute. This area may have been even lower because it was more concentrated. So to try to equalize that concentration, what I mean by concentration is an equal number of particles per unit area. Okay, what happens is water is going to go from this compartment, let me do it a different color, water is going to go from this compartment to this compartment until I finally, it says down here, say flow of water, it's going to go to until finally I get equal concentration levels on both sides, and that's called osmosis. Okay, osmosis involves water and water only in the human being. Water and water only. So if you hear me say osmosis, that's only pertaining to the movement of water. The other particles do not move across the membrane, only water. Okay, sort of like, going through a turnstile at the subway or the RTA, whatever the case may be. So osmosis occurs when the membrane is permeable to water, but not to other solutes. Because water is what? A solvent. It's only going to go according to from a lower concentration to a higher concentration, because it's a lower concentration because there's more water, has more water to give. So it's going to go through that membrane. Now this creates something, and you're going to hear this in physiology, called osmotic pressure. And osmotic pressure is the force of a solution containing the solute particles that cannot cross the membrane. Okay, so let me just say it in a different way. Okay, the force is proportional to the concentration of the solute particles that cannot cross the membrane, which means that the more concentrated something it is, okay, what does it want to become more dilute? More water. So it actually has like a sucking force. It's going to suck the water from the area that's more dilute to the area that's more concentrated. Okay, so it's almost like I look at osmotic pressure as like a sucking force to try to suck the water out of it. Okay, so the higher the solute concentration, the higher the osmotic pressure. Again, the solute is the particles. Higher the solute concentration means that that side of the membrane would have a higher concentration, be much more concentrated. So therefore, because it has much more concentration, it has a higher drawing or sucking pressure to pull the water for something that's more dilute over to the more concentrated side. Okay, and the pressure eventually becomes equal to the point where water will stop moving when we finally get that equalization, where the concentration on one side of the membrane is equal to the concentration on the other side of the membrane. Now the water levels will change. Okay, the water levels will change because if I start out with more particles on the one side, that water level is going to go up, because I'm going to be taking some of that fluid volume from the side that's more dilute and putting it across the membrane to the more concentrated area, so my fluid level is going to go up. Okay, makes sense? Now that makes sense when we look at the next thing here. Oh, osmosis. Let my people disperse. Okay, let my people disperse. Okay, thought that was cute. This has something to do with what we call tonicity. Okay, now things could be either what's called isotonic, hypotonic, or hypertonic. Okay, and what happens is tonicity as a word is the measure of a solution's ability to change the volume of cells by altering their water content. Okay, let's look at this over on the right hand side. I have a red blood cell and you're putting it in some water. Okay, now if the water concentration outside the cell is same as the water concentration inside the cell, where is the water going to go? And your answer to that should be almost automatically and immediately nowhere. It's going to stay put. So the cell stays the normal shape. It doesn't change size. It doesn't. So the cell's not going to gain water or anything like that. Okay. So that's called isotonic. Iso means the same. Iso as a word, iso means the same. So if something's isotonic, it means it has the same tone or the same concentration of that water on both sides. Okay, that's called isotonic. Iso means the same. Hypo means less than or lower. So let's take that red blood cell and stick it in a hypotonic solution over here, which means that the water concentration inside the red blood cell, okay, the water concentration inside the red blood cell is less than the concentration outside, which means that the intracellular material in the red blood cell is more concentrated than the concentration of the fluid outside. Okay, so in osmosis, what moves? Water moves. And water moves from more dilute to more concentrated. Makes sense. So if the inside of the cell is more concentrated and the water is more dilute, where's the water going to go? It's going to go from the outside inside the cell. As a result, the cell is going to get bigger and bigger and bigger and bigger and bigger and bigger, and it bursts. And that bursting is called hemolysis. Okay, so in a hypotonic solution, it means that the solution outside the cell is more dilute, okay, has a lower concentration of solute, higher concentration of solvent. In this case, solvent in the human body is what? Water. Therefore, what's going to happen is water is going to enter the cell, faster than it leaves, and the red cell is going to swell. It's eventually going to be burst. It's called hemolysis. On the other hand, if I have something that's hypertonic, okay, hyper means excessive or high. So now let's say I stick that same red blood cell in a solution where we have a higher solute concentration on the outside in the extracellular area and lower on the inside. Okay, so it's lower here, it's higher here. So therefore what happens is the inside of the cell is more dilute, the outside of the cell is more concentrated. What's going to happen is, again, with a solution with a higher concentration of solute than the cytosol or the cytoplasm of the cell, what's going to happen is it's going to start to suck that fluid out of the cell and bring it outwards into that solution because what can only go across? Water in osmosis. So water leaves the inside of the cell, the intracellular environment, goes through the cell membrane, goes to the outside. As a result, what happens? The cell is going to pucker up. It's called crenation and shrink and will eventually die. Okay, so basically this is just showing the way that water will move inside. Now, where is this important? This is important because if you look at IV solutions, I don't know how many people have had IV solutions. Guess what? All my IV solutions are isotonic. They basically have a concentration of water or concentration with particles from water that's similar to the concentration inside cells. So therefore, I don't have any movement of water back and forth between cells. You don't want to give somebody a hypotonic solution because what's going to happen is they're going to have all these red blood cells burst. Therefore, there's no ability to carry oxygen. And you want to stay at hypertonic because then all those red cells are going to shrink down. Okay, so this is basically has to do with osmosis, the movement of water, always going from an area that's more dilute because that membrane is semi-permeable, permeable only to water. Therefore, water is going to go from an area of more dilute area, which is more water, to an area of more concentrated, less water, to equalize that water concentration on both sides of the membrane. I think that's an important function to remember. Okay, and this just shows you. Let me get rid of all those pictures and stuff like that. Let me get rid of that so I can see this. There we go. Oh my gosh. So we have that hypotonic solution and it's swelling, we're going to burst. Okay. You look at the isotonic solution, it's pretty happy. Nothing's going on. If you look at that hypertonic solution, see it's shrinking. I'm shrinking. And it goes down to nothing. Okay. So anyway, that's a little about to try to explain what osmosis is. That's an important factor to remember. Okay. Now, active transport, like I mentioned, is different from passive transport. Passive transport does not require energy. Active transport does. So active transport says now I have a situation that requires energy to be able to get across that cell membrane. Where does that energy come from? It comes from ATP. We talked about that adenosine triphosphate, which is a high energy compound. What happens, I mentioned this before, that ATP has these 3 phosphate bonds in the molecule, and those bonds are under high energy. So what happens is to release that energy, you release one of those bonds, and energy is released to the outside, and that energy could be used to power something. Okay, and then that ATP then becomes ADP, or adenosine diphosphate, as compared to tri. Then it's going to actually go back, and they're going to make more energy by adding another phosphate to the ADP. Now it becomes ATP, and that ATP is stored for energy. Okay, so a couple examples of active transport. Okay, is what's called a sodium pump. Okay. Now, I mentioned before that like in muscle, cardiac muscle, nerve conduction, I have a chemical gradient with sodium on the outside, potassium on the inside, an electrochemical gradient with sodium on the outside, positively charged cation, and negative on the inside because of a lot of large negatively charged molecules like proteins. When that cell membrane of the skeletal muscle, the sarcolemma, is stimulated, it allows those sodium gated channels to open to let the sodium come in. But once the sodium gets in, you know what? It likes it in there. It doesn't want to leave. It stays. I'm going to stay for a while. My sister says, no. We have to get you outside because this may happen again. You know, we need you to be ready for the next time you need to come in. And, you know, so it's sort of like that. It's sort of like the relative that comes and says, hey, listen, do you have a place where I could sleep for a night? And you hope the next morning when you wake up, they're probably gone. Okay, you want to get them out, you hope that they're not going to be there for, a month later. They're saying, hey, you know what? I like it here a lot. I'm thinking about sticking around for a while. And you're, geez, what am I going to do? I can't even study my Bio 1221 or something like that. So anyway, what happens once they get the sodium inside the cell, it doesn't want to go. So as a result, what happens? We have to pump it out. It has to take that sodium and actively expel it and kick it out. Okay, sort of like get that bouncer and have that bouncer now throw somebody out of the club, and that requires energy. So to be able to expel the sodium from the inside of the cell to the outside, as well as to bring the potassium, because the potassium went out to try to equalize it, to try to be a temporary peacekeeper, the potassium has to be pumped back in, and that requires the use of energy. And again, that ATP then gets converted when it releases energy to ADP. Okay. So that's that. Also, we have that's called primary. Okay. Also, we have what's called a secondary active transport, and these involve moving substances across membrane with symporters or antiporters. And basically, what the symporters do is basically still uses energies, but moves two substances together in the same direction. Antiporters move two substances in the opposite direction. At least remember the primary active transport. I think that's really important. So basically, that's just looking at different types of active transport. Again, requiring energy to be able to use the energy being lost, the ATP, the phosphate being used as energy, resulting in the ADP. Okay, and obviously it requires energy to be able to transport things across membranes. Okay, so basically that's called active transport. Takes some power and some energy to do that. Okay, now again, like I mentioned before, now we've talked about a number of different things: diffusion, all kinds of osmosis, facilitated diffusion, active transport. We mentioned that some things are just too darn large to get out. Okay, and they're just too big. How are we going to get these things out? And a couple of them, one way we do it is transport them in what we call vesicles. Vesicles are like bubbles. Okay, and I mentioned there are two things: endocytosis and exocytosis, basically. And this is basically bulk transport, transporting large particles and large volumes of stuff inside and outside. And endocytosis basically occurs where what happens is now we have something where something will be it binds with a receptor on the outside. This is a bad picture. Okay, so don't look at the picture for now. It binds with a receptor on the outside. So material binds, and what happens is actually the cell membrane. Okay, once it binds to a receptor, the cell membrane will come and hook itself up around the outside of it. Okay, and as a result, what happens with that, it creates a bubble, on the outside. Okay, and then what happens is it actually is then able to take it and pull it in. Okay, so that bubble is formed on the outside. That bubble then pulls that whole material inside. So let me just see if I could draw a picture. I'm not really a good artist here, but here's the surface of the cell. Okay, and let's say what happens is I have this material that comes up in here. Okay, and it binds to a receptor on the surface of the cell. What's eventually going to happen is that cell membrane is actually going to come like this. It's going to start to wind itself around the outside like this and makes a bubble. Okay, makes a bubble and it creates a bubble inside there, and eventually this area right here is going to open and it spits that material on the inside of the cell. Okay, and that's called endocytosis. Okay, and that's what we see with something called endocytosis, bringing things in together. Okay, so endocytosis, this is the endocytosis right here. We talk about here again, we have a material. This is where it's bound, and here's the membrane, sort of like comes up around the outside of that material, binds it together, and extrudes it to the inside. Okay, that's what we see with that. Some things like, again, large particles, bacteria. We want to try to get rid of the bacteria, and we want the bacteria on the outside. So what happens? The bacteria will bind to the surface of the cell membrane. Cell membrane sort of like invaginates itself around it, makes that vesicle around the outside, brings it in, takes it to a lysosome. The lysosome destroys it and creates what's called a residual body that it'll eventually get rid of. Okay, that's that. So we're eating those things in endocytosis or phagocytosis in this situation. Okay, phagocytosis means eating something. Endocytosis means, again, bringing it in stuff. Okay, so that's a little bit. We also have what's called pinocytosis. Okay, pinocytosis is basically taking up something like a water and other material. They want to take up it, so it's mostly extracellular fluid with particles that are inside it. Okay, and basically it does this. It works the same as endocytosis. It creates a little vesicle around the outside, brings it in, and then once it gets inside the cell, it can be dispersed where it wants to go. Okay, so that's called pinocytosis. Endocytosis, pinocytosis. Pinocytosis is mostly fluids. Endocytosis are the particles and stuff like that that are on the outside. Okay. Drinking, like, and mostly water and stuff like that, mostly fluids. Exocytosis is just the opposite of that. Okay, what happens is when we have certain things that are made inside the cell and we want to get rid of them, you know, it's sort of like I know where I live, Wednesday's trash day. You take all your stuff, you put it out in your green bin or your blue bin, you stick them at the foot of your driveway, and the trucks come by and they empty your bins. Okay, and the same thing happens here. Now we have material that we don't want. Okay, and it could be like enzymes, hormones, mucus, other secretions, things, neurotransmitters that we could get rid of that get inside the cell, you know, other types of waste products and stuff like that. And what happens is the cell makes a vesicle around them. It sort of like brings these things together and forms a bubble around them. That bubble then migrates to the surface of the cell membrane, fuses with the cell membrane. Eventually the cell membrane opens to the outside. The opposite way that we see with endocytosis. So in endocytosis, something fuses to the surface from the outside, the extracellular side of the membrane, fuses to it. The cell membrane folds up around it, fuses, and pops it inside. Exocytosis, things inside the cell put together, put in a bubble. That bubble goes to the cell membrane surface, fuses to the cell membrane surface, eventually it gets extruded out like a mouth. Just spits it out. Okay, and that's called exocytosis. Exo from inside out, endocytosis from outside in. Large bubbles. That's how that's the endocytosis versus exocytosis. Okay, so that's the difference between the two. So what we've talked about today is I can't say it's like non-conquerable. I mean, everybody should be able to understand this stuff with a little thought process. But it is something that you need to know. These things are going to come up time and time and time again, because we're going to be talking about that cells need certain things. And how do I get same things inside cells? Why don't some things enter cells? Why do other things enter cells? You know, and if they want to, and if I want to get them, how do I get them in? You know, we talked about passive versus active. Passive doesn't require energy. Active does require. Passive forms, we talk about diffusion, simple diffusion, just going through the membrane. And certain things, we talked about factors which favor diffusion and factors which inhibit or limit diffusion. We've talked about facilitate diffusion, using some type of a protein to carry things across a membrane. We talked about ion pores that allow things, certain particles that normally can't diffuse, to go through a pore. You know, we talked about gated diffusion, which is basically a pore then that may open or close the gate. We talked about osmosis, and osmosis involves what? Water. Okay, it involves water, and that water moving across a membrane, a semi-permeable membrane, versus active transport, which means that what happens is now I need to use some type of a protein or some material to be able to get things across that cell membrane, okay, that requires energy. Now, not all proteins, again, facilitated diffusion may have a transmembrane protein, but doesn't require energy. It's just, I'll do it for nothing, you know, I mean, fine, you're a friend, I'll do it for nothing. Where, you know, you call out and you need somebody to clean out the junk in your garage, and yep, well, that's going to be that amount of money, you know, to do that. But it requires energy, it requires money. Okay. We also talked about how larger things, such as larger particles, bacteria, and bacteria are much larger than the molecules. Okay, bacteria you can see on a microscope. Okay, they're smaller than cells, but much larger than these molecules. And basically, to try to get larger things from the outside to the inside, we do that by endocytosis, with that cell membrane folding around the material. The material binds to a receptor on the surface. The cell membrane folds around it, creates a bubble around it, and that bubble then opens to the inside and deposits the material to the inside of the cell. Or exocytosis, when I have waste and other materials that are inside the cell that I just want to get rid of in bulk. okay? And what happens is we form a bubble around those things. That bubble goes to the surface, the cell membrane, fuses with that cell membrane, and once it fuses, it opens up to the outside and spits them to the outside. And those are things that, these are all things that will be, again, that we'll be talking about multiple times during the course of the semester that you need to know. So there's no better time than now to be able to get these down underneath your belt and to go over this lecture probably 7 or 10 times, okay? You don't have to do that, okay? But in other words, just to be able to understand how things move across the cell membrane is going to be critically important when we talk about a lot of things down the road, such as muscle contraction, nerve conduction, cardiac muscle contraction, all kinds of things like that, as well as things in the kidney. Kidney is all things moving back and forth all over the place, as well as filtration, which we have in certain patients of the kidney, okay? So anyway, hopefully this has been informative for you to give you a little bit of idea about how we get things inside and outside the cell. Say Uber can't deliver, so we have to decide a different way to get it inside, and there's a number of different ways, and hopefully you'll understand these ways. So hopefully this will make sense to you. If you have any questions, you could always ask me during our WebEx meetings or whatever the case may be, or in class. And other than that, be safe and be healthy. Okay, talk to you later. Bye-bye.